Hello, we are Sarrazin and Schmitzer from the Campus Institute Data Science at the University of Göttingen. In this video, we want to show you some of our research on optimal transport theory and its applications for data analysis and machine learning. We are currently working with the research group of Professor Tim Salted, which is also part of the University of Göttingen, to study the effect of Alzheimer's disease on the human brain. Professor Salted's group is specialized on phase contrast X-ray tomography. This is a modern technique to obtain high-resolution 3D images of sensitive cell tissue samples. In the animation, you can see a part of the human hippocampus at a resolution of approximately 200 nanometers. One can recognize individual cells and even their substructure, such as the nuclei. With such detailed 3D images, is it possible to learn something about Alzheimer's disease and how it affects the brain? In the future, this might lead to better diagnosis, treatment, or even prevention. But the quantitative analysis of such vast amounts of data will be very hard if one has to do it manually. Therefore, we are developing methods for automated analysis with computers. We will now present one such approach based on optimal transport theory. For our computational analysis, the first step will be image segmentation. This means we try to identify objects in the image with a distinct structure and boundary. This will typically be cell nuclei, other organelles, and blood vessels. By manually marking some of them, an algorithm can be trained to automatically segment the rest of the data. Here you can see a segmented tissue sample with nuclei and blood vessels. The red and the blue ellipses show zoom-ins onto smaller sets of nuclei and even an individual nucleus. The segmentation step in itself is an active field of research, with many scientists working to continually improve these methods.
Now that we have identified a list of cell nuclei, we can extract physically and medically relevant properties from each of them. This could be their volume, their density, or other similar properties. These extracted properties are often referred to as features. We can then think of every nucleus as a point, using these features as coordinates. Doing so for each highlighted nucleus in our list, the sample, which in this case corresponds to an Alzheimer's patient, can then be represented by the resulting point cloud in feature space. In the case of the brain tissue data, 5 features were used in total, but for simplicity, we will only draw 2 of them in this video. Let us make the same construction for each of our patients. Each one will yield a point cloud in feature space, which we have represented here in different coordinate systems and with different colors. Here we are interested in differences between the point clouds from Alzheimer's patients and those of individuals from a control group. We want to see how Alzheimer's disease alters the point clouds and be able to detect and interpret these alterations. To study these discrepancies, we need to be able to measure the similarity between 2 point clouds in feature space. The idea of quantifying these similarities is at the core of optimal transport theory. Optimal transport computes the most efficient way to move the points of one point cloud onto the points of the other, each red point traveling to a different green point in our case. In a slightly more technical level, we seek the pairing that minimizes the kinetic energy of the movement. The minimal kinetic energy is then called the optimal transport cost between the 2 point clouds. It defines a reasonable notion of similarity between them. In this first example, the 2 point clouds are fairly similar, giving us a transport plan represented by short arrows. On the other hand, when the 2 point clouds are much more dissimilar, the movement becomes longer, which is then reflected by a higher transportation cost.
Now that we can use optimal transport to compute distances between any 2 patients, we can think of each patient as a point in a more abstract patient space. This space is infinite dimensional and curved. Theoretically and computationally, it is perfectly possible to work with such complicated spaces. But for us humans, it is hard to visualize and interpret movement in this space. Fortunately, it is possible to approximate this complicated space with a low-dimensional flat space. This means that now we end up with a representation where each patient corresponds to a single point in a low-dimensional space. In our example, we used 4 dimensions, but we only draw 2 in the video. Recall that in the beginning, each patient was represented by a large 3D image with approximately 1 billion voxels. Now we are left with only a handful of very significant coordinates. This step is called dimensionality reduction and it is very important in machine learning to obtain results that are robust and can be interpreted by humans.
Now what can we observe in this simple representation? Let us start by colorizing each patient point according to their status, with samples from the control group colored in blue and samples from the group with Alzheimer's disease in yellow. Do we see a separation between blue and yellow point? Can we identify a direction in simplified patient space that corresponds to moving from healthy to increasingly affected status? In this case, it turns out we can. One can now ask how the brain tissue changes when moving along this direction. Let us recall that each marked point in this picture corresponds to one patient, which then corresponds to a point cloud in feature space. However, this correspondence is not specific to patient points, and any other point in this patient's coordinate space, a hypothetical patient if you will, corresponds to a hypothetical point cloud in feature space. One can now move this artificial patient in the direction of the control group or in the opposite direction of Alzheimer's disease. We can then simulate the corresponding evolution of cells in the feature space and study how their properties are affected. In the case of Alzheimer's data, it allowed to observe that the cells in patients affected with the disease on average became smaller, more dense, and with a stronger fluctuation in the cell density. This simple distinction between control group and the Alzheimer's patients could have been much harder to spot amidst the vast amount of data in the original segmented images.
Our project is only at the very beginning. The interpretation of the evolution of the artificial patients is still a largely open problem, but our preliminary results are very promising. You can find them in the reference paper. We are hopeful to extract more detailed information about how Alzheimer's disease affects the human brain in the future. In similar studies, we analyze how multiple sclerosis affects the brain or how COVID-19 affects the lungs. This research is made possible by the support of the Deutsche Forschungsgemeinschaft via the Sonderforschungsbereich 1456, Mathematics of Experiment at the University of Göttingen.